Hello everyone, thanks for watching. My name is Surabhi. I work at Centre for Advanced Imaging, Brisbane, Australia. I'll be talking about paper titled Echo Time Dependent Quantitative Susceptibility Mapping Contains Information on Tissue Properties Chosen for MRM Highlights. Quantitative susceptibility mapping is an emerging magnetic resonance imaging tool in assessing ion deposits, calcification, contrast region distribution. It is also extensively used in identification and categorization of multiple sclerosis, traumatic brain injury, and understanding physiological processes. Data were acquired from 32 channels. Magnitude data were individually processed for mask. Phase data were unwrapped and background phase was removed to calculate tissue phase. At the end, mask and tissue phase were used to generate combined susceptibility data. Figure shows the location of selected 10 brain regions for this study. Manual segmentation was performed for each participant for the selected 10 brain regions. Our work aims to explore information contained in the temporal trend by analyzing the mapped magnetic susceptibility as a function of echo time from gradient recall data acquired at 7T. We mapped magnetic susceptibility across the entire brain region of 5 participants from data acquired on a human 7T MRI scanner and extracted values for 10 brain regions. Results show that susceptibility values vary over echo time for caudate, internal capsule, red nucleus, and corpus callosum. The thin solid line represents individual participant data. The thick solid line represents the mean value computed at each echo time from 5 participant data. And dashed line represent one standard deviation either side of the mean. This study analyzes why the magnetic susceptibility values varies across echo times. Is it possible that this variance is attributed to tissue microstructure, and how can these results be interpreted? Similarly, a varying susceptibility response is observed in thalamus, pallidum, substantia nigra, putamen, fornix, and insula. From these results, we can conclude that there is a trend in susceptibility values with echo time, and the trend varies with the region selected and is similar in all 5 participants. We subsegmented substantia nigra, pallidum, and insula to study the influence of tissue cytoarchitecture variations on the TQSM curve. These regions are known to have different cell densities and cytoarchitectures, and the susceptibility curve depicts a different pattern for each case. The substantia nigra consists of compact and reticular parts, which have different chemical composition, neuronal density, and cell types. Pallidum's internal and external segments have different neuronal densities. The distinct deviation of the two curves of anterior and posterior insula may be caused by the presence of granular structures toward the anterior region and granular structures toward the posterior region. Each signal compartment of an image voxel is assumed to be describable using an amplitude modulated plane wave formulation. 3-compartment model have been used to describe the nonlinear nature of magnetic susceptibility response. We assume field changes as a result of microscale effects are contained in millimeter voxel scales. We opted for a robust method of optimizing for 9 parameters. Thereby, we implemented in MATLAB the global optimization method called differential evolution, which was used to fit the temporal evolution of QSM curves after values were averaged across all participants and each of the brain regions studied. All 10 brain regions were fitted onto a 3-compartment model. Table provides the result for 10 brain regions investigated by fitting the values used to parameterize the 3 susceptibility compartments. The adjusted R-square value is representative of the quality of fit achieved. For 9 out of 10 brain regions, the fitting process resulted in an adjusted R-square greater or equal to 0.9, implying a very good quality of fit. For pallidum, a good fit wasn't obtained. The reason that pallidum didn't have a good fit could be the inability of the three-compartment model to explain the trend in the TQSM curve. Secondly, the model assumes plane waves can be used to explain each compartment, which may not appropriately describe the signal behavior observed in experimental data. We investigated the temporal effects on quantitative susceptibility mapping. By studying 10 brain regions, we have been able to establish that the mapped value of susceptibility changes with echo time. All brain regions exhibited a good fit in three-compartmental model except pallidum. Microstructure of tissue potentially influences magnetic susceptibility compartments and the temporal susceptibility trend. We suggest that careful consideration be made to the acquisition parameters used to obtain quantitative susceptibility maps and how susceptibility values with echo times are used to assess brain changes. To conclude, our work demonstrates that microstructural influences possibly influence magnetic susceptibility at the voxel level both in gray and white matter. Decomposition of voxel constituents into meaningful parameters may lead to informative measures that reflect changes in tissue microstructure. Parameterization of image voxel susceptibility compartments provide a framework for new imaging biomarker developments in neurodegenerative diseases and disorders affecting the central nervous system. Thank you for your attention, and thanks everyone in Rutin's group at Center for Advanced Imaging.